координатор консилиума. И пока к нам все еще продолжают подключаться наши коллеги, хочу сказать, что, как обычно, наш вебинар проводится на двух языках, на английском, на русском. Пожалуйста, выбирайте удобный для просмотра канал и удобный для просмотра слайда презентации на русском или на английском. Запись данного вебинара ведется, и мы опубликуем все материалы данного вебинара, как обычно, на нашем веб-сайте, который мы будем транслировать в чате. Наш сегодняшний вебинар посвящен такой сложной теме. Я думаю, многие из нас в какой-то момент задумывались, что же делать с пациентами, которых не удалось излечить новыми схемами, новыми препаратами. Что же делать, если пациент изначально устойчив к большинству препаратам, которые на сегодняшний день имеются в стране. Для освещения и обсуждения этой сложной темы мы пригласили великолепных спикеров, так что пользуйтесь возможностью, задавайте свои вопросы во время вебинара и просим вас пользоваться функцией чата. То есть вы можете заранее задавать свои вопросы, и мы их обязательно озвучим в конце вебинара во время сессии вопросов и ответов. Ну и традиционно для начала нашего вебинара я передаю слово господину Аскару Yedilbayev.
Dear, thank you very much, Elmira. I mean, do you hear me? Yes, good. So first of all, I would like to welcome you on behalf of the WHO Regional Office for Europe, our team, on this 32nd webinar of the European Virtual Medical Consilium. Indeed, this is a very big achievement of having 32 webinars, and thank you very much for your active participation. And now we see that it's almost 150 participants and joining this webinar, which is showing that the high relevance of this regional platform that we are trying to use to deliver the most relevant information to you. So also, I just wanted to welcome you on the month of spring. And I'm wearing the symbol of Martișor, which is a traditional celebration in Romania and in the Republic of Moldova. And it's marking the arrival of spring on March 1st. And indeed, we are in spring is always bringing hope for the best, hope for the future. And I also would like to thank our speakers, Dr. Jennifer Furin and Dr. Ilaria Motta, for their participation and for their active engagement in the fight against tuberculosis and drug-resistant tuberculosis, which is highly relevant to the many countries of our WHO European region. And these days, indeed, a very important message that this webinar specifically is bringing is how to treat the most difficult cases with really severe resistance patterns of those clinically challenged. And this is the role of the Virtual Medical Consilium. And these days, it's very important to say that the multi-drug resistant tuberculosis and any resistance that is related to multi-drug tuberculosis is very important in the scope of the global health security agenda. And because resistance and tuberculosis does not recognize any walls, any borders. People are moving for this or that reasons. And not leading to the resistance is very important aspect, but also it's important how we treat those people who are suffering from these deadly forms of resistance. So thank you very much and welcome to the webinar.
Disease clinician and assistant professor at Harvard Medical School, who will present best practices for clinical management of tuberculosis with extensive drug resistance. Jennifer, the floor is yours. Dear colleagues, please ask your questions in the chat.
Great. Well, good morning, everyone, or good afternoon, or good evening. It's a real pleasure to be with you all today on this Consilium. And as Askar mentioned, I think with the start of spring, we would like to have some more hope for a change of season. I know it's been a very difficult time in TB, but I think we are all hoping that things will change. And I really appreciate all the work you all are doing. So this morning, I'm going to talk a little bit along with my colleague, Dr. Motta, about best practices in the care of people with drug-resistant TB that has extensive and expanded resistance. And so, sorry, as I get my slides ready here. So the objective, I just want to briefly talk about the global epidemiology of resistance to some of the newer and repurposed TB drugs, and then present some approaches or sort of common best practices that many of us are using globally to care for people with strains of TB that have these resistance patterns. So we know that these are very exciting times in the care of drug-resistant TB. Many of us have been working in the field for years, and we remember the old bad regimens that lasted for 18 to 24 months and contained many toxic drugs, including injectables. But for now, the WHO has recommendations for a majority of people to be treated with all oral shorter regimens, and most can be successfully treated in 6 to 9 months. I think most countries and most programs know about BPAL and BPALM, but there are now actual multiple options that exist based on high quality data from randomized controlled trials. And my colleague Ilaria is going to be talking about some novel compounds because there are new drugs coming out for TB, although many of them are being tested for drug-susceptible TB and treatment shortening. But it's really an exciting time to be in the field of TB. But we do know that even though there are standardized all-oral shorter regimens recommended for
пироральный режим лечения,
that there will still be patients who need individualized approaches. I think the first thing to point out is that although the trials often show treatment success rates ranging between 89 to 95% success, we know that in the real world, the success rates are not usually as high, in part because of the populations that enroll in trials, and in part because of the resources that are often available in trials to support people. In the trials, if a patient has toxicity or has to stop taking 1 or 2 of the medications, they're counted as meeting a trial endpoint, and they're often referred to the program for care. And you all are the program. So managing these individuals requires an individualized approach. And that we are going to see, and we're already seeing, resistance to many of these novel and repurposed agents. So baseline resistance or natural resistance always occurs. There's no drug in the world where there won't be strains of TB that show resistance. But what happens in therapy, if it's inadequate or the person isn't able to be supported through therapy, is that we select for these strains of drug resistance during suboptimal care. And that when we are unable to test for resistance at baseline, the selection of these resistant strains can be even more of a problem. And then these strains are often transmitted further in the community. So it's not surprising that we're seeing resistance to some of the group A and B drugs and the nitroimidazoles, but it does present a management challenge because all of the WHO recommended regimens contain bedaquiline. All of them contain linezolid, and most of them contain a 3rd-generation fluoroquinolone. So when you start seeing resistance to these agents, you can no longer use the all-oral shorter regimens. So how bad of a situation are we talking about? So when it comes to bedaquiline resistance, we have more data about bedaquiline resistance than we do about any of the other newer and repurposed drugs. And we know that it's a growing problem, both at baseline prior to treatment and bedaquiline resistance that develops during treatment. Most of the resistance is mediated through an efflux pump. So this is a pump in the mycobacteria that sends bedaquiline out of the cell. So bedaquiline is not able to be effective. And this happens when there's mutations in the mycobacterial genome in the RV0678 segment or in a segment called PEP-Q. The reason this is important is because there are many mutations that we can see, and so we're not likely to have a cartridge-based way to diagnose bedaquiline quickly like we have for rifampicin, INH, and the fluoroquinolones. Complicating this is that phenotypic testing can often have different clinical breakpoints, so we're not sure if lower doses of bedaquiline resistance are associated with the same clinical outcomes as higher doses of bedaquiline resistance. And one very complicated and unfortunate feature of bedaquiline resistant strains of TB is that there's almost always cross-resistance with clofazimine. There are some microbiologists who will say if you don't see clofazimine resistance, it's because you did the clofazimine resistance testing wrong. But if you lose the ability to use bedaquiline due to resistance, you often lose the ability to use clofazimine as well. And there are some regions in the world, particularly in Southern Africa, where there's a mutation in the RPOB region called the I491F mutation that's associated with bedaquiline-resistant strains. So how common is this? So multiple studies have shown that baseline between 2 and 3% of newly diagnosed patients will have strains of TB with bedaquiline resistance, but this is as high as 7% in some cohorts and in some settings. And then during treatment, we see between 2 and 3% of people who are treated with bedaquiline will develop resistance during the course of therapy. There are some cohort studies, however, that show in certain populations, bedaquiline resistance is much more common. So a study was done in Cape Town that showed that among people who still had a positive culture at 4 months and were being treated with a bedaquiline-based regimen, 8% of them had bedaquiline resistance at baseline when people went back and looked, and 47% of them had acquired bedaquiline resistance over the course of the treatment. And risk factors for this include baseline fluoroquinolone resistance, prior exposure to clofazimine, and fewer than 4 effective drugs in the treatment regimen. In terms of linezolid resistance, we don't have as much information about this, but linezolid resistance, it usually happens later in the course of therapy, and most of the resistance is due to mutations in the mycobacterial ribosomes. So some people believe it takes a lot more to develop resistance to linezolid. A meta-analysis that was done showed a pooled frequency of linezolid resistance of around 4.2%, but higher rates have been seen in some settings. So in an MSF cohort from Mumbai, linezolid resistance was seen in 19.7% of the strains that were making people sick in that study. And linezolid resistance may be more common in people in settings where there's broad antibiotic access, because linezolid is commonly used to treat other types of infections. In terms of delaminid resistance, there's very little data and experience. We know that delaminid is a prodrug and has to go through multiple metabolic steps to become active, and any mutations in the pathway can confer resistance. In a study done in China, 4.4% of the strains of TB had baseline resistance, and trials show that people acquire resistance between a rate of 1 and 2%, and there may be cross-resistance with protomanid, the other nitroimidazole. And then in terms of protomanid resistance, we know very little about this, but it is also a prodrug like delaminid. In the Xenix trial, 1% of people had strains of TB that showed protomanid resistance. And we do suspect that in lineage 1 strains of Mycobacterium tuberculosis, there may be more protomanid resistance as there are higher MICs to this drug. So when we have, as Askar said, extensive drug-resistant TB or expanded resistance to any of these agents, we know that we see poor clinical outcomes. So some cohort studies have shown that treatment success rates when there's resistance to these agents are around 30 to 40%. So this is discouraging and disappointing. And this is a problem in many settings because there's limited access to DST. So when patients present at baseline, they may not have drug susceptibility testing done. And so it's only when they're doing poorly or not responding to treatment that DST is sent. And the drug susceptibility testing often takes several weeks to come back. it's an interesting problem to have theoretically, but in reality, it's a terrible problem to have. And many of us know that patient populations are presenting for care now. Those of you who are working, taking care of patients have probably seen patients with these types of resistance. We need to think about how to manage these individuals. Currently, there are no trials to guide us. And so many people are just doing the best that they can to care for these patients when they come to their clinics. So in order to help support each other, in order to help support patients and programs, a group of us got together and formed something called the BETTER Project. And BETTER stands for Building Experience Treating Tuberculosis with Expanded Resistance. This is a volunteer group of frontline providers, impacted communities, TB survivors, TB programs, and civil society organizations that have come together to share best practices around how we're caring for individuals that have strains of TB with this type of resistance. And really, the goal is to try to provide some common principles and guiding principles for treatment in the absence of formal recommendations. We know we're not going to have formal recommendations on caring for people that have these strains of TB for a while because there really isn't any evidence for the WHO to make these formal recommendations. But in the meantime, rather than have everybody as an individual or an individual program try to come up with care plans for these individuals, we thought it would be good to join together in a community of practice. And many of you on this call are actually part of the BETTER Project and have contributed to this. I want to just mention the terminology expanded resistance. So we came up with this term because it covers types of TB that have monoresistance to bedaquiline, linezolid, clofazimine, or the nitroimidazoles. We certainly prefer the WHO terminology of extensive resistance, which would include resistance to a fluoroquinolone and another group A drug. But even if an individual doesn't have extensive resistance according to the formal definition, and they have resistance to one of these agents, we know that they'll need an individualized care plan. So the issues that we cover, and we have a field guide that was released in December, and that I'm happy to share with the group here. We cover issues around optimizing drug susceptibility testing, around informed consent and shared decision-making, how to design regimens, how to have holistic packages of support, so it's not just around the regimen that we design, care of special populations. We have principles around pre-approval access to novel compounds, which my colleague Dr. Motta is going to talk about in more detail. We talk about an approach to managing household contacts who've had exposure to these strains of TB, as well as toxicity monitoring and management and operational research considerations. So I'll hit some of the highlights from the field guide here. So in terms of optimizing drug susceptibility testing, ideally everyone would have rapid drug susceptibility testing at baseline to all the agents in the regimens they're going to receive. We know that that's not happening, and that most people have baseline testing to rifampicin and many to the fluoroquinolones, but in many settings, drug susceptibility testing to bedaquiline, linezolid, the nitroimidazoles is not being done. So we need to advocate for more investment in this. There's a trend sometimes in TB to want a universal regimen where we don't have to do drug susceptibility testing, but really, we need to do drug susceptibility testing, and we need to keep advocating for this. There are new techniques coming out in terms of targeted next-generation sequencing and whole genome sequencing that should advance this field. But in the absence of universal DST, the risk groups that should be tested are really anyone who's had prior exposure to these drugs, anyone who has a positive culture at month 3 or more, who's on one of the all-oral shorter regimens, anyone who's had treatment interruption, anyone who's a contact of individuals with these risk factors. And some providers would say people with extensive disease, so bilateral disease, cavitary disease, should have drug susceptibility testing done to bedaquiline, linezolid, and the nitroimidazoles. One of the issues that's very important when treating people who have strains of TB with extensive and expanded resistance is informed consent and shared decision-making. This is a topic that's often new in TB, not the informed consent part, because we're used to having people sign consent forms, but shared decision-making. Because we don't have a lot of evidence on how to treat people with these strains of TB, it's important that we understand patients' goals and values when they're coming into treatment, right? We're not going to ask a patient to design a regimen, but in areas where there's uncertainty or we're not sure what the best thing is to do, it's important to talk to people about their values, their goals, and their preferences in treatment. And that's really what shared decision-making is all about. And when we don't have good evidence, when we're talking about using regimens that may not have very high success rates, it's really important to approach this in a partnership with the patients. And so we're not going to ask them, oh, what regimen would you like? That's not what shared decision-making is. What shared decision-making is that we understand what the patients value. So for example, if we're going to have to hospitalize them to administer a carbapenem, we have to understand how that's going to impact their lives and look at other options that may be available if they can't be hospitalized for long durations. So there are different ways that people with expanded and extensive resistance come into care, and it's important to think about those because they often require a different approach. So we've been using bedaquiline, many of us, in settings for almost 10 years now. And so there will be patients who come into care who have a recurrence of TB and they had a remote prior exposure. And so that's one group of patients. Another group of patients are the patients who are currently on therapy with one of the WHO recommended all oral shorter regimens, and they're not doing well. So they may still have a positive culture at month 3. They may not be responding clinically. They may have had some struggles with adherence, particularly in the early part of therapy. And these are individuals that we know have a high likelihood of having expanded or extensive resistance, and we need to think about how to individualize their regimen. Then there's a group of patients who may be on these all oral shorter regimens and they're doing great, but you sent a baseline resistance test that comes back 8 weeks later and shows drug resistance. So what do you do in those settings? And then there are contacts of all of these individuals, and they may require slightly different approaches when we think about them. When we're designing regimens for these populations of patients, either with known or possible resistance to the agents like bedaquiline, clofazimine, linezolid, the nitroimidazoles, or the fluoroquinolones, one thing to think about is, can we use a standardized regimen? And the answer is no. So there are very few regimens that have been tested that don't contain bedaquiline, linezolid, or fluoroquinolone. So the NTB trial that was done, NTB regimen 5 is a bedaquiline sparing regimen, although I would point out it does contain clofazimine. So this was a regimen of delaminid, clofazimine, moxifloxacin, and pyrazinamide. And this regimen in the modified intention to treat population did show non-inferiority, but in the per protocol population, it did not establish non-inferiority. So this regimen is not recommended, and people on this regimen who were not cured actually developed additional resistance during the course of their treatment. There is the MDR END regimen, which was a trial that was done in South Korea. That is a 9-month regimen of delaminid, levofloxacin, linezolid, and pyrazinamide. That regimen, when compared with the standard of care, showed non-inferiority, but it really should only be used in people with non-severe disease, and in general is not recommended. It hasn't been reviewed by the WHO. But there really are no regimens that are standardized that are non-severe. And then I just want to mention here, there is a WHO recommendation for using BPAL for patients who have strains of TB with fluoroquinolone resistance. But there's some recent data coming out from the END TBQ trial that suggests that maybe people with strains of TB that have quinolone resistance and severe disease should get a longer individualized regimen. So just be careful in using standardized regimens in people with whose strains have quinolone resistance, but certainly in the setting of bedaquiline resistance, linezolid resistance, nitroimidazole resistance, there's really no standardized regimen that can be used. So you're going to have to design an individual regimen for most of these patients. And in our best practices field guide, we put together some plans for designing an individual regimen. It's very similar to the WHO principles for individual regimen design. You need to use 4 to 5 likely effective drugs, and probably you should be using 5 drugs unless the person has very minimal disease. Most of these regimens will contain cycloserine or taururzidone. We're reaching now for the older drugs, things we haven't used in a while or we were glad to be rid of, things like PAS, ethionamide, and amikacin, as well as we're often recommending that you use the carbapenems with clavulanic acid. Really, the duration of this regimen should be 12 to 18 months after culture conversion, and you will be using injectable agents and drugs that have higher rates of toxicity, so that's something to keep in mind. It's important to use a combination of both bactericidal and sterilizing agents. So you need drugs that will kill rapidly multiplying mycobacteria, as well as more quiescent or latent forms. The bactericidal drugs are very important in the early part of therapy. We have a list of drugs that we consider more bactericidal or more sterilizing, but I do want to point out that defining which drugs work in which way can be tricky. And so just keep that in mind. Again, we recommend a stepwise approach as the WHO. In step 1, you choose as many group A drugs as you can. In step 2, you choose additional oral agents that have bactericidal activity. In step 3, you choose additional oral agents with sterilizing activity, so drugs like PZA, clofazimine, although again, clofazimine may not be useful in people whose strains of TB have bedaquiline resistance. Then we add amikacin if it's susceptible and able to monitor for hearing loss, and add a carbapenem plus clavulanic acid. Then you consider other backup drugs, things like PAS, and then consider drugs available through pre-approval access or compassionate use, which Dr. Motta will speak about in just a couple minutes. There are some drugs you can keep around even if you're uncertain about whether or not the drug is effective. So this may be because there's a mutation that has uncertain significance, or it may be because the drug is highly effective and you don't have drug susceptibility testing, so things like bedaquiline, linezolid, and the quinolone. But it's important to talk with patients and let them know you're keeping a drug that may not be effective, and it's important to consider things like pill burden. There is some experience using higher doses of some of the drugs, and this may be because there are 2 different breakpoints seen in phenotypic testing, or because of the idea that a higher dose may be able to overcome some of the mechanisms of resistance or have better penetration into some sites of disease. So you could consider using higher doses of bedaquiline. So it's a 500 milligram loading dose for 14 days, followed by 200 milligrams daily. Higher doses of clofazimine, so 300 milligrams daily, and then higher doses of the quinolones, linezolid or moxifloxacin. But you really have to monitor for toxicity if you're going to use these higher doses. As we know, a higher dose is more likely to be associated with toxicity. It's important to consider ancillary medications and support, things like nutritional support, corticosteroids for meningitis or pericarditis. And then there's also some interest in these strains that have a lot of resistance in using calcium channel blockers. The efflux pump that mediates resistance can be blocked by calcium channel blockers. So there's some thought that this may be a useful adjuvant therapy in patients whose strains have a lot of resistance, although there's not a lot of experience with this. It's important for holistic packages of support to be given, not just a new drug regimen. Thinking about the location of care, mental health services, counseling, nutritional support, financial support, talking to patients around transitions to care if they're going in or out of the hospital, palliative care, and then post-TB lung disease, because many of these patients often have had TB for prolonged periods of time. We need to think about children, pregnant people, people who are in congregate settings like prisons, homeless shelters, migrants, because these populations may need specialized dosing or maybe at increased risk for strains of TB that have expanded or extensive resistance. And then finally, I'm going to skip this slide because Dr. Motta is going to come on next and talk about pre-approval access to novel compounds. And then I just want to mention that toxicity monitoring and management is really important. We're in a stage now where we're reaching back for many of these older, more toxic drugs to treat people that have strains of TB with extensive and expanded resistance, and that we really need to monitor them and include toxicity management in their treatment plans. As we're treating people who have these strains of TB, it's important that we share our experiences with each other and with the WHO so they can make recommendations and we can move beyond best practices. But it's good to use common data elements and to consider, having prospective observational and operational research cohorts. So the Better Project is continuing our work in spite of the cuts to funding. We're a volunteer community and we're open to anyone who would want to join us to learn, to share their practices. And then as data emerge, we'll continue to update our field guide with each other. So that's all I had to say. Sorry if I ran a little bit over on my time, but my email address is here. You can reach out to me at any point. I'm always happy to talk about TB, to talk about the care of patients. And I just want to acknowledge the strong contributions of the many colleagues who are part of the Better Project, including many of you on this call. So thank you so much. And I'm going to stop sharing my screen. And I think we'll take questions at the end, but I know some questions may have also come up in the chat. So.
Thank you so much, Jennifer. Большое спасибо, Jennifer, за прекрасную презентацию, обзор.
Thank you very much, Jennifer, for an excellent presentation, for your overview. I know it is very early for you in the morning, but your presentation has been absolutely interesting. Special thanks to your group, the development of the field practice book for treatment of patients with drug resistance. I have shared the link to your guide, and you can find that in the chat. Dear colleagues, you can download it and use it in your practice. And now I would like to give the floor to the next presenter, Ilaria Motta, who is a physician who will tell us about the new TB drugs and practices of pre-access. Please, Ilaria, the floor is yours.
Thank you. Can you just confirm you can hear me well and see my slides? My slides are in English, so you can select on top of the Zoom if you need in Russian or English, just because it took me a while to look at the Jen's ones in English. So, okay, so my name is Ilaria Motta. Thanks for the invitation. I work with MSF Access to Products for Healthcare, formerly known as Access Campaign. I'm also part of the clinical trial unit at UCL here in London, and I'm a part of United for TB consortium investigating new chemical entities for TB. So the talk will be divided into parts. The first bit will be presenting some new compounds that are now around phase 2 of development, and the second half will be based on MSF experience about practices of pre-access, and some of the compounds presenting the first part of the talk could be good candidate for potential.
Sorry, Ilaria, we don't see your slide. Could you share once again?
Sorry about that. Yeah. Can you see now?
Oh, great. Yes. So now we see.
Okay. Yeah.
Please type in the chat if you have some problems with the slide. Thank you so much, Ilaria.
Yeah. Sorry about that. Yeah. So I said these two parts. So let's start with the first one about the novel TB compounds. I'm sure you've seen these slides many times. And as Jen said, is quite an exciting time to work in TB, as there are many compounds under development. And I'm gonna focus more on this phase 2 part. And we will start with the first one that is one of the most advanced is quabodepistat. Quabodepistat is a compound from a new class of agents, and the class is inhibitor of DPRE1 enzyme, and so is around cell wall biosynthesis. This compound just completed phase 2. The developer is Otsuka, and you may have known that this was under investigation in the pan-TB trial consortium in a 4-month regimens for drug susceptible TB. But was presented at the Union. So I took this slide from the Union conference platform. But the pan-TB trial was early stop. I'm not gonna go in much details, but just to present the trial, the first stage that was undergoing at the time was the stage 1, where the 2 arms with this compound quabodepistat were given together with delamanid or pretomanid, bedaquiline, and sutedzolid for 4 months, and the control arm with the standard of care for drug susceptible TB. And the main objective of this trial was to find a short regimen. So that was designed to identify a 3-month regimen. And the enrollment started in 2023, but was stopped in the summer of 2024 with around 100 participants enrolled, and was early stopped because there were no safety concerns, but because were not found enough data to support the design of a 3-month regimen, because at month 2 and month 3, the sputum culture conversion were lower than the standard of care, and so this was decided to stop earlier. But I just also around this quabodepistat, always from Union, was also presented the result of this phase 2B2C trial. This study had only 3 compound, quabodepistat, bedaquiline, and sutedzolid, and was a dose finding optimal dose for quabodepistat between 10 milligrams and 90 milligrams. To let you know, in the pan-TB, quabodepistat was given at 30 milligrams. And this study was again for 4 months treatment in drug susceptible TB versus the standard of care. So the results were presented at the Union, and here the primary endpoint was the safety and the sputum culture conversion at the end of the treatment at 4 months. And actually here, the result presented met their primary objective, because was not inferior the pooled data, so for all the different doses versus the standard of care, the different proportion of the sputum culture conversion were within the non-inferiority margin chosen. But was also presented the outcome and the relapse at 12 months post-randomization. And actually compared to the control arm, there were more relapses in the investigational arms. This was not one of the objective of the study, but was presented. So to keep this in mind about the potential for reduction of the duration of treatment. And this is about quabodepistat, as said, is one of the most advanced in terms of safety. Also in this study were not found any particular warning. Another compound from the same class is BTZ-043. The developer is LMU, Munich University. Doesn't still have a name as quabodepistat, so it's still anacronymous. There's been a publication early this year about the Panacea, one Panacea study on the EBA that we know, and also this compound appears to be safe. There were some signal on the hepatic metabolism, and also this study is undergoing phase 2b/2c trial for a shortening in a drug susceptible TB as part of the United for TB Consortium, and I'm part of this study. Just to disclose any involvement. So I put this slide just because you may have heard that this new class, so the inhibitors of this enzyme that is involved in the synthesis of aranobinogalactan, is not just quabodepistat and BTZ. There are the two compounds in development in this class. There is the potential for cross-resistance with mutants of RV0678 that you know, as Jen presented, as resistant to bedaquiline. It seems that the mutants in this gene confer a low-level resistance to this class, but is not yet known the clinical significance of this finding, because at the moment there are not enough data to say that if that can be overcome by increasing dose. We really don't know, but I want to put, because there's been reports about that. Another compound is gamfeborol by GSK, a different class, a protein synthesis inhibitor, has been published some studies on EBA. Also at the Union has been presented those findings before, and at this Union also with different arms that contain bedaquiline, delamanid, and or just bedaquiline or just delamanid. That study also has other arms with other compounds, companion compounds, like the BTZ was mentioning before, and linezolid is also under assessment in the under United for TB Consortium, again for drug susceptible TB, in a shortening of the duration of the regimen. Telacebec. So Telacebec, I couldn't find recent news about this compound. That we have some initial data from phase 1 and phase 2. Now the compound is held by TB Alliance, but no further information on the development in more advanced study. This compound inhibits the ATP synthesis with a different mechanism of action. And also we know about linezolid and all the drawbacks for toxicity with the long use. There are under development other compounds from the same family that inhibit the protein synthesis. In this slide, you can see that there are at different stage of development. Probably the most advanced are the delpazolid and sutezolid. Tedizolid, for example, some of them were not just developed for TB. It comes from antigram positive therapy, but were under investigation for TB. Some of them, like TBI, is pretty early in development, phase 1. And in terms of, it's going to be look at the safety. The padrisim have good safety as sutezolid, and also with potential drug-drug interaction. And the main things for these compounds is to look at their toxicity, and it seems that they have less or none myelosuppression, as we see with the use of linezolid. So this potential good compound to be used for longer period if needed. Then, as last new compounds are these one, are from the same class as bedaquiline. The one on the left is a bit earlier in development. The one on the right is most advanced, are really promising because of both their potency. So it seems that could overcome resistance to RV067 and 8 mutants, and so having lower MIC for in these mutants compared to bedaquiline. The one on the left, as I said, is early in development. The one on the right has been studied in phase 2 studies, NC009 by TB Alliance. Both of them are by TB Alliance, and NC009 is looking at 3 different doses of this compound together with predominant and linezolid versus the standard of care for drug susceptible versus BPAL. And so and also in terms of toxicity, it seems to have less impact on QT interval prolongation. Now there is quite high hope for this compound, TBA 876. And this is about the compound. Now I'm gonna move more on the second part of the talk, that is about pre-approval access. And again, this is more from MSF experience. I want to thank my colleague Christophe Perrin that gave me his slide that is presented, some of them in other talk. So why is important to consider the compassionate use for in TB. Of course, like Jen said, we have really patients that don't have other option. So of course, this would be really important to give hope, and also actually have early access to some settings, and then once WHO recommended for some of these compound, we will have also add additional data and be used earlier as a programmatic in a programmatic setting. Of course, not always is is felt positively because the innovators that are working on this compound, they have all their reason to be cautious because trying to protect their new compound, and also not having enough data in terms of efficacy in different type of population or different duration. So it's a kind of balance that, and it's good that the pre-approval access is done in conjunction with different actors, and the compounds that can be considered are the one that have at least the phase 2 data, and are some of them that I previously presented. So what MSF did in the past under the NTB project was trying to have a modus operandi in different settings, in different projects, both from MSF and PIH. And this was approved by the MSF Ethical Review Board. The patient is on board with this decision, so is well informed. And the use of this new compound with limited data from safety and efficacy, and is required a written consent from the side of the physician, is the one responsible to present a request to the medical committee that I'm going in a second, and follow the protocol or the documentation given for the use of that specific compound, and also agreeing in reporting safety findings as per protocol agreed, with in a timely manner. In the previous experience that worked very well, there is a medical committee for compassionate use that review all the requests for cases that could be eligible for the use of these compounds, and this medical committee are composed by experts in the field, not just from MSF, and then they agree, so for a consensus, they agree and propose give some recommendation and indication, and report to the ethical review board by MSF. Also is very important, and this is really considered essential both by MSF and by innovators, the presence of strong pharmacovigilance. In this experience, MSF pharmacovigilance unit was the one that was dealing with all the reporting and safety reporting, and also was doing the training in the setting. Okay. Depending on the setting, there could be different requirement, and so basically would be good to know if from the audience here, you know, if something I'm saying maybe it does not apply to your country, but is really always good to have a consultation with the National TB program about the value of introducing as a pre-approval access a new compound. A candidate for this use should have at least phase 2 clinical data, as said before. And the protocol is going to be reviewed by the ethics review board, usually at the, depending on the setting, but usually also the single hospital that is using the drug should be reviewed as well. The informed consent procedures, and reviewing through the National Regulatory Authority, the pharmaceutical quality documents. And also in terms of for importing the drugs, having also this outline with the authority, competent authority. In terms of the innovators, the innovators are, of course, the ones that are providing the compound and also give all the information available and to the local authorities, and also to the clinical side, the information about the safety, and give also feedback on a series of adverse events, and also data that they are accumulating in other studies, ongoing study. The innovator has the final say about the use and availability of the compound, and so we review the protocol and to decide which type of patients, as Jen was saying. We have a patient with bedaquiline resistant. We have still patient that, due to the expanded resistance, still go under injectable. So this could be potential good group of participants to introduce one of these new chemical compounds. And also the sponsor, the innovator, usually ask to the practitioner or the hospital where they work to sign an agreement, and can put some requirements on the pharmacovigilance safety reporting. For example, in the experience with delamanid, was a requirement by Otsuka to have a PV unit. So in terms of putting all this together, of course, it's difficult to do this completely for free. So funding is needed, and the area where are more needed is to put together the medical committee. Was really important in the past experience. Actually, there is still ongoing a medical committee for difficult cases in MSF from the NTB project. They are still reviewing complicated cases, so it's not something that got completely lost. But of course, in case of this new project together with Better, there should be a need to strengthen that and streamline again. And this medical committee would have a strong connection with the innovators and with the physician. We were having together with Jen and others colleague some initial conversation with different innovators, and there is interest from their side to have compassionate use, let's say, platform where more than one of new compound are combined together and have kind of compassionate use regimen. Of course, it's a bit tricky for different reasons. The first one is because they are a different stage of development. And so if Quabodepista is more advanced, it's not the same for others. But I think it's a good momentum to keep this conversation alive, with the aim to have more than one drugs that we can use in these expanded resistant strain. And then comes from other challenges in terms of procuring the drug. GDF, well, considering all what's going around, but in the past was available to help and support for the supply. Also because some innovators are not big companies, GSK and Otsuka, but some like BTZ from LMU could not have the fund to provide compassionate use treatment completely for free, also due to manufacturing capacity. and so there could be some adding cost in that part. So this was pretty much what I wanted to present today, but I'm happy to see what are your your interests, your questions, and thanks for your attention. Over to you.
Thank you so much, Ilaria, for this excellent presentation and great overview of what TB compounds are in the pipeline and what could be available by the countries. We have quite interesting discussion in our chat, but before we proceed, I would like to clarify, maybe I just wanted to clarify if indeed, first of all, national TB programs have to decide if they really want to proceed with the compassionate use or not. And when they feel that they are ready, there is a demand, they are committed, should it be their, should they approach to the innovators or the manufacturers themselves directly, or maybe I've got it wrong, but as far as I understood, MSF could serve as a partner in getting these drugs, in getting the access to compassionate use medicines. So what should be the next step of the country when they think that they are ready, they want? Whom should they apply to?
Yeah, maybe I can start, and then I will definitely hand over to Jen, because I think this is also really in line with the objective of a platform like Better. This was from the experience with NTB project, so where there was already an ongoing project and MSF with the experience setting. But as I said, we're not just the medical committee, for example, was not just MSF. The first step, of course, like mapping the landscape and be aware what is happening is the first things, but also would be good for each country to know what they are, their national policy, their requirement in terms of documentation, if it's need a protocol, because Jen can explain better than me, as she has much more experience, but according to the setting could be different. What are the requirements, if it's considered under a research protocol, this, even if it's not a research, the compassionate use, and then the liaison. I think this is what MSF together with other organization can be, and also this can be also felt as a trusted strong structure for innovators to give a kind of homogeneous approach in different setting, and because using this compound in this programmatic setting, but with a protocol and a safety reporting pathway, we can actually also have data to share with innovators that would be difficult in under a normal clinical trial due to stringent eligibility criteria, etc. So actually are information that are useful, and at the same time we offer an option for the patient. But I want pass to Jen about what could be the next step. Move for Better.
Yeah, thanks, Ilaria. I think you said it perfectly. Right now, while there are exciting compounds, we still don't have access to the ones that are furthest along, which would be quabodepestat. So we're advocating with the company right now to try to come up with a structure for that. But I think in the meantime, what countries and programs should do, as Ilaria said, is understand your framework for how you can use these drugs when they're not yet approved in country. And most countries had experience doing this with bedaquiline and delamanid. So reviewing what that looked like and being ready so that, you know, as soon as the company allows this pre-approval access or compassionate use to go forward, the countries that you're ready to receive them. As Ilaria said, there will probably be an application process. We'll probably have an independent medical committee to review the patient's histories, because you don't want the drug company doing that, and then have a way to ship the product to the country. But then the country has to know, how are you going to use it? How are you going to monitor it? What are your pharmacovigilance systems? What kind of data are you going to collect that's required by the company? What kind of data are you going to collect that's required by your own country? But again, most countries did this, right, with the Daquilene. And so I think there's a structure in many places. It just looks different for everybody. And then one of the things we are working on right now for countries that need an operational research protocol is to have sort of a standard protocol that we can share with countries, so that you don't have to develop that all by yourself. There will be common elements that go in any sort of protocol. So that's something else that Better is working on, and we're happy to share as we move things along. Over.
Thank you so much, Jen. Thank you, Ilaria. So we have 25 minutes for question and answers. And let me read aloud the questions that were already posted, and Jen already kindly answered, but probably it would be more convenient for all participants if we read them aloud. So Jennifer will have an opportunity to answer once again. One of the questions is about the BPAL regimen effectiveness in the patients with additional fluoroquinolone resistance. So Jen already mentioned that one of the risk factors for developing additional resistance is less than 4 drugs in the treatment regimen. So what is your opinion about using BPAL regimen in patients with fluoroquinolone resistance?
Yeah, so this is a great question, and it has a little bit of a complicated answer. So what I think is important for people to know is that to date, BPAL has not been tested in a formal trial in people who have strains of TB with quinolone resistance that contained a control arm, right? So if you look at the Practical trial, if you look at the NIX trial, and if you look at the ZNIX trial, right, those included people that had strains of TB with fluoroquinolone susceptibility and people that had strains of TB with fluoroquinolone resistance. So there's only been one trial done that enrolled people with strains of TB that had fluoroquinolone resistance, and that's the END-TBQ trial, right? So that trial was done, and the results were reported in Bali. And the reason that I mention this is that it's important because those other studies were not solely focused on people who had strains of TB with fluoroquinolone resistance. They were likely underpowered, right? And so there are conclusions that were made about the small number of people that had fluoroquinolone resistant strains in those studies, but those were not studies dedicated to looking at people who had fluoroquinolone resistant strains. So the END-TBQ trial was done by MSF PIH IRD. It was solely focused on people with strains of TB that had quinolone resistance, and it randomized them to receive either a standardized regimen of bedaquiline, delamanid, linezolid, and clofazimine for 6 to 9 months, depending on how extensive their disease was, or an individualized longer regimen. So in people who had minimal disease, so not bilateral and no cavities, that standardized 6 to 9 month regimen was great. It was found to be non-inferior to the individualized longer regimen. But in people with extensive disease, so they had bilateral disease or cavitary disease, that standardized 4-drug regimen was not found to be non-inferior. So the success rates were high, right? I just heard Carol Mitnick give a talk on this, and she obviously answers this question better than I do as one of the PIs on the study. The success rates in both arms were high, right? So in the standardized regimen of bedaquiline, delamanid, linezolid, and clofazimine, the success rate was 86%, right? So if you didn't have a control arm, you would say, great, it's a great regimen. But compared to the individualized longer regimen in people with extensive disease, the standardized regimen did not perform as well. In addition, the people who did not have successful treatment developed resistance to those drugs. And so this was a trial that was just focused on people whose strains had quinolone resistance. And I think it certainly calls into question whether or not we should be giving BPAL to people who have strains of quinolone resistance and who have extensive disease. If you have minimal disease, it's fine. But with extensive disease, I think a longer individualized regimen that contained bedaquiline, delamanid, linezolid, clofazimine, but it also contained taurursodone or cycloserine, that regimen seems to do better. That evidence has not yet been reviewed by the WHO, so we need to see what those recommendations show. And I know that countries have had good experience with BPAL, and this isn't to say don't use it. It's just that in a controlled trial where there was a comparison arm, the shorter all oral regimen did not do as well. So in patients with extensive disease, it's worth thinking about whether or not BPAL is the best regimen for them. More will be revealed as WHO reviews that data, but that's what the NTBQ trial showed.
Thank you so much, Jen. Ilaria, do you have something to add?
No, I think Jen covered well. Yeah.
Okay. Thank you so much. So the next question was from Liga. Quite practical question. Actually, we sometimes face with situations when the MDR-TB patients return to treatment, either with relapse or with failing regimen, and the repeated DST show susceptibility to the medicines that were used in the regimen. So what is your experience? What is your practice in designing the new treatment regimen for these patients? Would you rely on the DST in that case?
Yeah, you know, I think it as with any test, right, DST is not 100%, right? So with phenotypic DST, we can sometimes see different breakpoints. The microbiologists will even argue sometimes to say, well, this isn't really resistant. And some will say, no, it is resistant, or there's high dose and low dose resistance. And then we know that with the whole genome sequencing, right, there are a number of mutations that have uncertain significance. We don't know, right? And so I think interpreting drug susceptibility testing can be a challenge, and it's good to call the lab and talk to them and ask them these questions. If they say the strain is susceptible, but you have some uncertainty about that, right? We treat a patient, right? We don't treat a drug susceptibility result. And so again, this is where the concept of shared decision-making comes in. You know, and then you have to take into account the clinical scenario, right? If I have someone who was treated with BPALM and they struggled, you know, in the first few weeks, and there was a lot of non-adherence, and they managed to finish the regimen, but they come back into care later, I'm going to be more worried, even in the setting of susceptibility to those drugs, than I would be if someone was able, you know, to take the regimen fully and well and converted their culture quickly and had minimal disease. So I will often design an individualized regimen for those patients. I will usually include drugs like bedaquiline and linezolid because they're associated with lower mortality. But using a standard regimen in a patient who's had previous treatment is something where I consider the clinical scenario as well as, you know, sort of what their prior treatment looked like, and often come up with an individualized regimen. But Ilaria, I don't know if you want to add anything.
Thanks. Well, it would be then more about the different, the Liga was asking, WGS, etc. I'm not a microbiology. I see in the call there is Claudio Kozer. I don't know if you want to say something, but yeah, I think depending also for which drug we are talking about, the DST, as Jen mentioned before, bedaquiline can have multiple mutation, and we still don't know for some of them the significance. So I think I agree that keeping like bedaquiline, but not counting completely effective, just because we are not sure, I think is something that makes sense. And at the moment, and I don't know if we'll change in the future. I think individualized seems the right approach. Yeah.
Thank you so much. Thank you, Jen. Thank you, Ilaria. There is a question that probably Askar may answer as the representative of WHO. The question was about Pretomanitu, what a drug group it belongs to, ABC.
Yeah, I mean, the question is, thank you for the question. I think the Pretomani does not belong to any of the group because it was reviewed by the guideline developing group and recommended by the WHO as part of the regimen of the BPAL regimen and BPAL MOXIE regimen. So that's Pretomani does not belong to any. Thank you.
Yeah, thank you so much. And Askar, there is another note from Andrei Dudnik, I think. Yeah, he mentioned that in Spain, recently oral carbapenem, parapenem, and intravenous ceftazidime, avibactam, were included in the guidelines of TB management. Do you have any information from WHO side on the review of this medicine as TB drugs? As far as I know, I didn't heard that kind of information.
If as of what the information is currently available in the currently approved guidelines, carbapenems, imipenem, and meropenem are part of the Group C drugs for the long individualized regimens. So I mean,
yes, but ceftazidime, probably not yet.
Not yet.
We didn't hear about that.
I haven't heard. Thank you.
Yeah, and ferropenem, which is an oral carbapenem, that has actually been tested against TB in an early bactericidal activity trial, and it did not show any early bactericidal activity. So I don't know why ferropenem would be included, but the EBA trials on ferropenem showed no EBA. Thanks.
Thank you so much. And I think that one of the last questions, but it is not directly related to the topic of our webinar, but it's related to the development of resistance. So Andrei Dadu asked a question, what is your thoughts on the potential of and risk of development of resistance, fluoroquinolone resistance during the preventive TB treatment with levofloxacin?
Yeah, it's a question that always comes up when we talk about preventive therapy, right? Is are we causing resistance? And so there have been numerous trials with drug susceptible TB, looking at the development of isoniazid resistance after preventive therapy, looking at the development of isoniazid and rifamycin resistance after preventive therapy. And those trials show there's no risk for developing resistance. In both the TB CHAMP trial and the VQuin trial, which are the two trials that support the use of levofloxacin preventive therapy for drug-resistant TB, and which formed the basis of the WHO recommendation that levofloxacin be administered to household contacts of people who are newly diagnosed with drug-resistant TB. There was no risk of fluoroquinolone resistance in either of those trials either. And then there have been a number of operational research studies that are smaller cohorts and hard to make conclusions about efficacy. But we can see if any of the participants in those observational and operational research trials developed fluoroquinolone resistant strains of TB after preventive therapy, and they did not, right? So this is a theoretical concern that we often have, but so far the data do not support that. I think Andre was also asking what to do in terms of preventive therapy for household contacts of people with strains that have bedaquiline resistance or linezolid resistance or quinolone resistance, right? And right now there's no evidence to support any recommendations. The Phoenix trial, which is a large multi-center trial that's being done by the AIDS Clinical Trials Group, is looking at delaminid for preventive therapy. So we may have some information on that. It's a large trial. It just finished recruiting. And then I know there are individual experiences where bedaquiline has been used for preventive therapy. I think there's a cohort from Vladimir, Russia. So these are some potential options which we may be able to think about using in the future. I do want to mention the RATIONS trial, which is a trial that was done in India in drug-susceptible TB that looked at giving nutritional support to household contacts. So it was a cluster randomized trial that looked at nutritional support for household contacts exposed to TB and found the provision of nutritional support for 6 months decreased the likelihood of developing TB. So at the very least, if you have household contacts who are exposed to TB that has extensive or expanded resistance, I think giving that household nutritional support can decrease the risk. And then obviously close monitoring. So
thank you so much, Jan. Thank you very much, dear speakers, honored speakers, for the excellent presentation, for the excellent discussion. Thank you very much, participants, for your active participation and for the active discussion in the chat. Thank you for all the links that you provided here. We will definitely benefit from them. And I would like to give the word to Askar to close this webinar. Askar, the floor is yours.
Thank you. Thank you very much, Elmira. I would like to thank our speakers, Jennifer and Ilaria, for this opportunity to, share the information on the management of those patients, of course, whom we, in our countries, started observing. And this is not something new, but physicians and health providers are dealing with this kind of people every day. So this information and the approaches that you provided and shared beyond of what WHO is recommending, of course, is really good. So we, as the Virtual Medical Consilium in the WHO Euro, are providing and sharing, using the opportunity of sharing the most latest information on what is WHO recommending, but also given an information on any projects that are happening and the trials are happening in the globe for the physicians to consider, because the WHO European region is indeed, despite the fact that having the highest burden of drug-resistant TB, is also in its advanced stage of knowledge and experience in managing of patients with drug-resistant tuberculosis. This is not something new that we learn, but of course the questions are arising, and they're arising every single day. So I would like to thank you very much for providing this useful information, for sharing it. I think that the manual that you developed, Jennifer, with the team of colleagues, will be shared for the consideration by our physicians, and we will continue bringing this up for the discussion at the Virtual Medical Consilium. And its experts are open for the providing support. I think there is more to go, and especially with the members of the Virtual Medical Consilium itself on how to address that. But again, more evidence is needed because it's really promising that there are medicines in the pipeline, and we have to be prepared for once they are reviewed by the Guideline Development Group and WHO and recommended that countries are introducing. But at the same time, if there is a possibility for the clinical trials or projects like using these medicines, for the within their capacities, so countries can also explore for the best of the people's lives. So I would like to thank you for this opportunity, and also remind that that we should be also working hard of preventing the development of resistance and treating patients and offering them the best care